board. So yeah, so what we're going to do, I've never heard it say that before, is just look at the different parts of the digestive system, and we'll look at some of the names that we need to, definitions that we need to know, have a bit of a recap over enzymes. I know we've talked about enzymes before in the past, but we've sort of talked about them in sort of a general process and what enzymes are, how they work, the theory behind them, and then we're sort of bringing back some of that GCSE enzymes and digestion knowledge as well. So yeah, so one of the keywords that we have to know is lacteals. Lacteals are the lymphatic vessels found at the centre of villi. So the lymphatic system is all to do with secretions of hormones and glands and enzymes and lots of different secretions. So they're vessels that are found at the centre of our villi in our small intestine. And then we have the lumen. Lumen is a word that you probably come across again before. Lumen you will probably remember from when you've done blood vessels. So the lumen is that cavity, hollow part of the structure. So for example, you have a lumen in capillaries, arteries, veins. You also have a lumen inside your intestines. So that's a word that we will start to use quite a bit. Micelles. Micelles is something we've come across before. Micelles are our emulsified lipid droplets. So when our lipids are emulsified, they turn into these droplets that are in this circular spherical shape, and we make a micelle. Now, we won't have come across this before, which is chylomicrons. Now, chylomicrons, these are the products of triglycerides, and this is when they associate with cholesterol and lipoproteins. So if you have a look at the name again, I'm trying to go backwards and I don't think I can do it for some reason. It's triglycerides when they're associated with cholesterol and lipoproteins. Exocytosis. We spoke about exocytosis a couple of weeks ago. It's a type of transport mechanism, so similar in a way when we've talked about diffusion, osmosis, active transport. We have exocytosis and endocytosis. And exocytosis is movement out of a cell. We're exiting the cell, usually through vesicles. And we also have the ileum. Now, the ileum is another name for the small intestine. We have the colon, which is the large intestine. So they're two different words that mean the same thing. So colon, large intestine, ileum, small intestine. Enzymes, which we've come across before, which are our biological catalysts, and they are produced by a gland. Hydrolysis, which we've come across loads, which is a breakdown of a compound by chemical reaction with water. And then we have assimilate. And assimilate is when our nutrients in our food are taken into the cells of our body, so we assimilate them inside our body. So a couple of other things we're going to go through as well. Monosaccharides. Monosaccharides we've talked about before, carbohydrates. So they're a class of sugars. They cannot be hydrolyzed to give a simple sugar. So these are monosaccharides. We can't make them any more basic. Disaccharides. And these are class of sugars whose molecules contain 2 monosaccharides residues, and then obviously a polysaccharide is one that contains 3 or more monosaccharides. And we can actually break these down. So then we can have a monomer. Obviously, a molecule bonded to other identical molecules to make a polymer. Therefore, a polymer is the opposite. It's a molecule that large number of similar units that can be bonded together, which we can then break down into monomers. Lipids, being fats. Peptides are a compound of 2 or more linked amino acids. These are smaller than proteins, remember. A glycosidic bond, we've used glycosidic bonds a few times when we've spoken about glucose and monosaccharides and carbohydrates. So these are a bond which links a carbohydrate to another molecule, which may be another carbohydrate, and then emulsification, which occurs in digestion, which is our breakdowns of fats into smaller molecules. So we're going through the stages of digestion. We start off with a physical breakdown, and this is often, you occasionally can get asked to write a long answer question about digestion, and this is often the stage that's forgotten about and missed out by most people, is that initial physical digestion. So chewing our food, we've got a mechanical physical breakdown of our food there, and we also have a physical breakdown in the stomach as well. So that's always really important to remember, that we chew our food, and then our stomach also breaks the food down more as well. We then also have chemical breakdowns, which obviously involves all the enzymes. And this is where we're going to hydrolyze our large insoluble molecules into smaller soluble molecules. So this is the part that's carried out by enzymes, and the enzymes function by hydrolysis. So they work using hydrolysis. And hydrolysis, remember, is when we use water to break chemical bonds down. So the enzymes function by hydrolysis, and they use water to break these chemical bonds down to make these polymers into monomers. And remember, enzymes are specific. So the enzyme used to break down a carbohydrate isn't going to be able to break down a protein. Enzyme for a lipid isn't going to be able to break down a protein either. They're all substrate specific. So we have lots and lots and lots and lots of different digestive enzymes. You'll have probably heard of quite a few in the past, things like carbohydrates or amylase or lipase. They're the main ones that you've probably come across at GCSE, and we're going to have a look at them and a few more. So there are stages of digestion. So the physical one we don't really talk about too much. It's just obviously the chewing, the churning in the stomach. But then we have our chemical digestion, and this is the really interesting part. So chemical digestion, we have carbohydrates. As you can probably guess from the name of carbohydrates, these are enzymes that break down carbohydrates. If you remember back to when we've talked about enzymes before, they often end in ASE. And what's really quite nice at the beginning, we have carbo high. We just get rid of the drates in the end and put A's, so carbohydrates. These are enzymes that break down carbohydrates. We also have lipases. Again, they're enzymes that break down lipids. We have proteases. These are enzymes that break down proteins. So, carbohydrates, they break down a carbohydrate into a monosaccharide using hydrolysis. Lipases, they break down lipids into glycerol and fatty acids by hydrolysis. So remember, lipids are made up of glycerol and fatty acids, so they become, so we get the two different products breaking down lipids. And then proteases, we break down protein into amino acids by hydrolysis. So this is quite nice. If some of these polymers, monomers stuff you can't really quite remember from right back at the beginning, it might be a good point at some point to review those notes from the beginning. But if you can remember them, that's absolutely fine, and we'll just carry on with this. You don't need to worry about looking back over it. But yeah, basically this is what's happening here with these enzymes and how they break them down and what they're breaking them down into. It's basically the polymers into the monomers. Always using hydrolysis. And a reminder, hydrolysis is a breakdown of a compound by a chemical reaction with water. So let's look at carbohydrates to start off with. So digesting carbohydrates, basically, carbohydrates are a very, very big, large molecule, and we need to break that down into a smaller one. So when we digest carbohydrates, we break the large molecule down into a smaller one. We then break these smaller molecules down into monomers. So we have a really large molecule, we break it down into a smaller one, and then these smaller molecules are then further broken down into the monomers. And then different enzymes will act at different stages of the digestion of carbohydrates. It's not as simple as you have this enzyme, it breaks down carbohydrates, it works at this point. So actually, as you know, our saliva makes, our saliva doesn't, but in our saliva we have enzymes, We also have enzymes in our stomach. We also have enzymes in our small intestine. We're at different points of the digestive system. Obviously, if all the enzymes worked in our mouth, we wouldn't need any in our small intestine, but obviously they break things down differently, so they work at different stages. And enzymes need to be added in a correct sequence. So if we had an enzyme that should be found in the small intestine, found in our mouth, it wouldn't be able to work because our substrates that we would have in our mouth wouldn't be the right ones. It wouldn't be specific enough. It wouldn't have been broken down small enough for them to work. So they have to be added in the right stages for this to actually happen. So this is an enzyme, a carbohydrase enzyme that you've probably come across before, amylase. Now, what we're going to start doing now is talk about the stages of which they occur and how we break them down. So amylase, it's made in two places. It's made in the mouth, so in the salivary glands, and in the pancreas, in the pancreatic glands. Amylase, what amylase does is it hydrolyzes glycosidic bonds of starch. So it breaks down those glycosidic bonds that you find in starch. So remember, a glycosidic bond is a bond which links a carbohydrate to another molecule, which may be another carbohydrate. And so in starch, you've got lots of glycosidic bonds between each, it breaks them down. So this produces the disaccharide maltose, which is a 2-linked glucose unit. So we are not at a monosaccharide at this point. So this is still too large for us to digest straight away. So we've broken down our starch into maltose using amylase. So that's our first enzyme that comes across. Then we have maltase, which you can probably guess. Maltase is the enzyme for breaking down maltose. So maltase is a disaccharidase. So a disaccharidase is an enzyme that breaks down disaccharides, and this is produced in the ileum. Reminder, the ileum is a small intestine, and it hydrolyzes maltose into an alpha glucose. I'm really annoyed that this has an F spelling for alpha. I guess you can spell it that way. It should be a PH. I borrowed this from somebody, and now that's really annoyed me. So that's basically what would happen with the carbohydrates, carbohydrases. We have to break down into amylases and then into maltase. Amylase breaks them down from starch to maltose, and then maltase breaks down maltose to alpha glucose. And alpha glucose is then soluble and then can be assimilated inside the body. So an example of an exam question that you could get about this would be something along the lines of this. Describe the role of the enzymes of the digestive system in the complete breakdown of starch. This would be a 5-mark question. So if we go through the answers, this is just roughly what they give you, and we can start to form that into a full answer. So basically, you need to mention amylase. So basically, you could say something along the lines of, and I'm going to actually write this on. Let me annotate text. No, I want to draw actually. Okay, so so describe roles of enzymes. So right, let's mention that first enzyme. We can bullet point it if we want. Amylase. Where is amylase made? You can actually include that as well. So you'd want to write in full sentences. I'm going to bullet point this sort of make the point. But yeah, so we'd have amylase, which is made in our salivary glands. So it's found in the mouth and in the pancreas. What does amylase do? Well, amylase breaks down starch into maltose. It does this via hydrolysis. Then the second enzyme we have is maltase. Maltase is obviously found in the ileum. What does maltase do? Well, maltase breaks down maltose into alpha glucose. How does it do that? Well, it does this by hydrolysis, and what it actually does is it hydrolyzes glycosidic bonds, and actually the top one does the same as well. So glycosidic bonds are hydrolyzed, they're broken. So you can have that for both. So that will give you those 5 marks, and we can double check it with this. I say this, but it's not working. Here we go. Amylase says amylase is starch to maltose. Then we have maltase, which is maltose to glucose. Mark for saying hydrolysis, and it's the hydrolysis of glycosidic bonds. And you wouldn't be penalised in this question for the incorrect site for digestion or incorrect site of enzyme production, because it's not actually asking for them. Lots of people will include them in there, and that's absolutely fine for you to do that. It's just a reminder that you wouldn't get penalised if you either didn't include them or put the wrong ones. Right, let's clear all those drawings so we can see this one. Here's another exam question. The diagram represents part of the human digestive system. The organs are labelled A to F. Give the letter of the organ that produces amylase. Okay, so we've talked about amylase. Amylase is obviously produced, it's found in salivary glands and produced in also the pancreas. So we would, oh my goodness, we would say C for pancreas. And then give the letter of the organ that produces maltase. Well, we said that was found in the ileum. So that would be our small intestine, which on here is E. Struggling to see where that was on there then. So yeah, that's E. Again, another example of the exam question for this. Maltose is hydrolyzed by the enzyme maltase. Explain why maltase catalyzes only this reaction. And this is then when we're going to start joining in some of our enzyme theory and actually our information about digestion and enzymes. So here, you basically want to talk about the fact that its specific active site has a specific shape. So here we go. Our active site of our enzyme has a specific shape. You can say it's got a specific tertiary structure. You can also say the active site is complementary to a substrate or a maltose, and something along the lines of a specific shape. The active site has a specific shape. Active site is underlined because you must include the words active site if we're talking about its specific shape. So active site has a specific shape, and it's the active site of the enzyme, which is really important as well. So you need to make it clear that the enzyme has a specific active site shape, not the substrate, which can happen or it can be quite unclear in questions. You can't say they have the same shape as each other, because obviously it's a lock and key induced fit theory idea. So actually they wouldn't have the same shape. They'd be like a jigsaw pattern. They have specific shapes that are complementary to each other, but they won't be the exact same shape. Therefore, if it has a specific enzyme, active site shape on the enzyme, only maltose combined fit in. Now, you need to make sure you're saying maltose in this point and not maltase, because obviously maltose is a substrate. You could get away with saying only the substrate can fit, but that substrate is maltose. And when they fit together, they form an enzyme substrate complex, so an ES complex. So our processes. So let me just close that a second. Our saliva mixes with our food. Our saliva contains salivary amylase. Starch gets broken down into maltose via hydrolysis using aided by the amylase enzymes. Its salts help maintain a neutral pH. This is really, really important because the optimum enzyme pH for salivary amylase is neutral. It's a pH 7. So salts help to maintain that neutral pH. Then what happens is the food will then enter your stomach. The acid, the stomach acid that you've got in there, actually denatures salivary amylase, and that's to prevent further breakdown of starch. Now there is there is reasoning behind this. Our acid denatures those salivary amylase. So often we just say, oh yeah, the acids help break down the food, but actually if salivary amylase only works at pH 7, the second the food enters the stomach, we have acid in there, the salivary amylase will no longer work there. Then our food passes to our ileum. In our ileum, food is mixed with the pancreatic juice. In the pancreatic juice, we have pancreatic amylase. So it's still amylase, still breaks starch down into maltose, but it's just made in the pancreas rather than the salivary glands. And so the pancreatic juice contains pancreatic amylase, and this ensures the remaining starch is hydrolyzed to maltose. Alkaline salts produced by the ileum and the pancreas ensure optimum pH for enzyme activity 7 again. So again, we have often people will call them bile salts. You might have heard them referred to as bile salts, and that's what this is. These salts help maintain that pH 7. Then our ileum muscles push the food along, and remember that's that peristalsis movement where it's sort of almost like a Mexican wave. One bit pushes and it pushes the next bit, peristalsis, and the lining of which produces maltase, a disaccharide. So the lining of the ileum, so the actual small intestine, the ileum, produces maltase, a disaccharide, which is another type of enzyme. Maltase is released into the lumen of the ileum. Reminder that lumen is that empty hollow cavity of the small intestine, the ileum, but it also forms part of the lining to the ileum as well. So we have maltase on the lining of the ileum and into the lumen of the ileum as well. So this is how it gets its name, membrane-bound disaccharidase. So it gets referred to as membrane-bound disaccharidase because it forms part of the lining of the ileum as well as it's being released. So that forming part of the lining makes it a membrane-bound disaccharidase. What maltase does is it hydrolyzes maltose into alpha glucose, which would have originally come from our starch. So it's that last process of breaking down our starch into alpha glucose. We have to have amylase in between and then maltase. So then we get another example of an exam question. So we have the diagram shows, I don't know what it's saying it's showing, because I can't see what it says it's showing. I presume it says an epithelial cell. Let's have a quick look. Let's click on mouse. Click here. Let's just have a quick look at what it's actually saying. Epithelial cell from the small intestine. And it wants you to name organelle Y. Where is Y? So obviously Y, it's just testing your organelle knowledge. Y obviously is a mitochondria. There are a large number of organelle Y in this cell. Explain how these organelles help the cell to absorb the products of digestion. So then you have to think, okay, why would this epithelial cell need lots of mitochondria? Well, okay, mitochondria is the site of respiration, produces ATP. ATP is conversion of energy. Why would we need energy? Oh, well, okay, so we're getting food is passing into us, into the cell, and so we're having assimilation happening, but that would happen by diffusion, surely, yes. But remember when we went back to talking about diffusion and active transport, eventually that concentration gradient will get to a point where it's at equilibrium. So we don't have a high concentration gradient on the outside than the inside of the cell. So diffusion will stop. When diffusion stops, digestion switches to active transport. What do we need for active transport? We need ATP, adenosine triphosphate. Where is ATP produced? In the mitochondria. Would it then be useful for the cell to have lots of mitochondria? Yes. Why? It makes lots of ATP. Why is that good? It allows active transport to take place. Why do we need active transport? Well, when our concentration gradient gets to the point of equilibrium, diffusion can't happen any longer, and we don't want to waste all that food and all the nutrients that we've bothered to break down. We've spent a lot of energy eating the food, spent a lot of energy making those enzymes. We've broken it down to this point, and then we're going to go, oh no, we'll just sort of get rid of, once we get to halfway point, we'll get rid of it. No, we switch to active transport. So mitochondria, and then cytophobe aerobic respiration, ATP production, energy release, active transport, transport against the concentration gradient. So sucrose and lactose. Let's have a look at these two. Now, these are disaccharides, sucrose and lactose. Right, let me say this properly. Sucrase breaks down sucrose into glucose and fructose. Lactase breaks down lactose into glucose and galactose. So often a lot easier to see than say, because we know the differences between sucrose and lactose. They're our substrates, and sucrase and lactase are enzymes because they have ASE on the end. So let's look at lipids. So actually what we do when we digest lipids is we don't just go straight in with lipase. What we actually do is we split them into micelles first. So some of those bile salts produced by the liver ensure that we can do this. And this is emulsification. So this increases surface area to ensure lipases work more effectively. If we literally just had like one, it increases our surface area so lipases work better. We want, again, that's the usual part of biology, we want a larger surface area, and so things can work more effectively, more quickly. So what are triglycerides and what are monoglycerides? Well, we know triglycerides, one glycerol to 3 fatty acids, monoglycerol, glycerides, 1 glycerol to 1 fatty acid. They're broken down by lipases, and lipases are produced in the pancreas. And what happens here is they hydrolyze the ester bond in triglycerides. So in triglycerides, we have the glycerol, 3 fatty acids. We have that ester bond between the fatty acids and the glycerol. Lipase breaks down, it hydrolyzes that ester bond in the triglycerides. So triglycerides are lipid groups bound by ester bonds of linked molecules of 3 or more fatty acids molecules and 1 molecule of glycerol. So this one's forming fatty acids and monoglycerides, and monoglycerides consist of 1 fatty acid molecule and 1 molecule of glycerol. So actually when we break a triglyceride down, we're very used to, from like back at GCSE, of saying, oh, we get 3 fatty acids and 1 glycerol. Technically, we get, let's say it is a triglyceride with 3 fatty acids, we get 2 fatty acids and 1 monoglyceride molecule. So 1 fatty acid still stays attached to our monoglyceride. So I'm going to have a look at what endopeptidases are, exopeptidases are, and dipeptidases are. So these again end in ASE. So they're more examples of different enzymes. So endopeptidases, what these do is they hydrolyze, again, using water to break down chemical bonds, the peptide bonds between amino acids. Quite clearly in the name. So we know it's ACE, it's an enzyme, so it hydrolyzes. Peptide bonds. Okay, these are in a specific place. So these are the peptide bonds between amino acids in the central region of a protein molecule, forming a series of peptide molecules. So endo in the center of it. Exopeptidases hydrolyze the peptide bonds on the terminal amino acids of the peptide molecules formed by endopeptidases. So in this way, they progressively release dipeptides and single amino acids. So basically, endopeptidases come in and they get in the central region and we start breaking it down into smaller peptide molecules. Exopeptidases come in and they're getting those terminal ends of the amino acids and breaking them down. So what we then end up getting is we get a release of dipeptides and single amino acids. Single amino acids, we don't need to do anything else to, they can just get assimilated. Then we have dipeptidases, which you can probably guess break down dipeptides. So these hydrolyze the bonds between the two amino acids of a dipeptide. Dipeptidases are membrane bound, being part of the cell surface membrane of epithelial cells lining the ileum. So this is happening basically, we're about to get absorbed, we're about to get assimilated via diffusion, but they can't because they're not broken down small enough. So we have it as a membrane bound enzyme. It gets broken down at that point. So a typical exam question for this, endopeptidases and exopeptidases are involved in the hydrolysis of proteins. Name the other type of enzyme required for the complete hydrolysis of proteins to amino acids. That would be dipeptidases. You could say membrane-bound dipeptidases as well, that's fine. So just explain why the combined actions of endopeptidases and exopeptidases are more efficient than exopeptidases on their own. Well, basically, endopeptidases, it's just asking you what the difference are, what do they do? So endopeptidases hydrolyze those internal peptide bonds, whereas exopeptidases remove those amino acids, hydrolyze those bonds at the end. So more ends or increase the surface area. So we're basically increasing the surface area. If we're using, if we increase the surface area, we're able to get in, break them down more. Makes it a lot more efficient, a lot more quicker. If we just use endopeptidases on their own, it would take a long time to work our way out. Similarly, if we use exopeptidases, it would take us a long way to work our way in. So carbohydrates, they are broken down by salivary amylase, which is made of the salivary glands, and that produces maltose. We also have pancreatic amylase, which is found in the pancreas, and that, well, made in pancreas, and that produces maltose. Then we have maltase, which is produced in the small intestine, the ileum, and that produces alpha glucose. Proteins, we have three enzymes, for, well, three examples of enzymes here for them. So we have pepsin, trypsin, and peptidases. Now, all those exopeptidases, endopeptidases, and dipeptidases all come under the peptidases bracket. Pepsin is found in the stomach and breaks down peptides. Trypsin is found in the pancreas, and breaks down peptides. And peptidases is then when we have in our small intestine break down our amino acids. Fats, lipase, produced in the pancreas, and it makes fatty acids and glycerol. So, and look at the different parts of the digestive system now. So we've got the duodenum and the ileum. So the first 20 centimeters of the small intestine is known as a duodenum. This is why we start using different terms like the duodenum and the ileum, because actually these are the different parts of the small intestine. So rather than just going, yeah, I've got small intestine, it's massive, where absorption happens, we're now being more specific. What happens at each part? Because actually in the different parts of the small intestine, different things happen. So first 20 centimeters of small intestine is known as the duodenum, and this is where the secretions from the liver and the pancreas are added. So that's where those pancreatic juices are added. So that is this first 20 centimeters here, the duodenum. And then we call the rest of the intestine the ileum, and that's about 5 meters long. So technically the whole thing is the small intestine. The first small section is the duodenum, and then the rest of the small intestine is the ileum. This is the ileum is where the final processes of digestion occur, but this area is highly specialized for absorption with several features which contribute to its efficiency. So all those adaptations we're going to talk about allow the ileum to be very good for absorption. You don't find them in duodenum because the duodenum is intended just for releasing secretions. So we have our small intestine here. On our small intestine, we have villi. These are our villi. we have these infolds into our small intestine, increases our surface area, increases the rate of absorption of food. Then we have, if we have a look at this villi, we have a basement layer. This basement layer is called the submucosa. Submucosa is where some of these mucus layers come out from. We have our mucosa layer above that and muscularis above that. If we then look in at this smaller section, we then have our microvilli. So we can have it coming up to each microvilli. We have a capillary network. That's really, really important because we're going to be wanting the transfer of all these nutrients going into our blood. So we have a capillary network. We have lymph vessels there. And so we call all of this section the villus. We also have a vein, which will carry our nutrient-laden blood to the body via the liver. And we'll also have arteries, which will carry our oxygen-rich blood to our villi. If we then looking even more closer, we have these epithelial cells that make up our microvilli. That contains our nucleus, our cell membrane, and then our microvilli on the top. So absorption. So most, and again, this is how it goes for most things. Most absorption takes place in the small intestines and large intestines, specifically small intestines in the ileum. So mainly digested food in the small intestine and water in the large intestine. And again, it's the word mainly. And there can be bits of food absorbed in the large intestine, bits of water absorbed in the small intestine, but generally that's the way around it works. It's long, okay? So it provides a large surface area for absorption to occur. So the small intestine, large intestine added together, it's long. It means we've got a large surface area. And we have a large number of villi in the mucosa, increasing the surface area again. So we've got that increased surface area for absorption. So if we have a look at our ileum, we have the lumen of our small intestine up here. There we go, that's our lumen. And here we have our villi, and then if we look closer, we have the microvilli on top of our villi. So the villi possess smooth muscle fibers, and what these smooth muscle fibers are able to do, they're able to contract and relax. And the contraction and relaxing motion of them mixes up the contents and brings the epithelial cells into contact with the digested food. There are microvilli on the columnar epithelial cells to further increase the surface area. So an epithelial cell is cells which layer the outside of an organ. And an endothelial cell, cells which provide the inner layer of an organ. And that is the difference. So epithelial, outside layer of the organ, endothelial, inside layer of the organ. So state which structure is responsible for most of the nutrient absorption. Well, that would be the ileum, small intestine. State where water absorption occurs in the gut. That would be our large intestine or the colon. Explain the adaptations of the gut for efficient absorption. So with its surface area. Large number of intestinal villi increase the surface area along the inner mucosal surface of the intestine to increase the rate of absorption. So basically, lots of villi increase our surface area. So microvilli. Microvilli are 1 micrometer long and 0.1 micrometer wide. And a single villus has lots of microvilli on them. So villi, each villus has an extensive capillary network that we just spoke about. So why we want that? That's so that our food can be absorbed and transported away quickly. So it means that we maintain this really large concentration gradient. If we didn't, we'd actually hit equilibrium very fast and nothing would really happen, and we'd have to rely on a huge amount of active transport for digestion to take place. If food just sort of sat on the other side of the intestine for a while, we'd sort of reach an equilibrium part and nothing would happen. So we'd have to use a lot of active transport to try and get the rest of those nutrients through, which would be probably not very energy efficient. We also have a lacteal in each villus, and that's where our fats are absorbed. This is the lacteal in there. The fats don't go into the blood vessels. So goblet cells, I don't know if we've spoken about goblet cells before, but goblet cells, I think we have when we spoke about the lungs, but goblet cells sit in between epithelial cells. They're goblet shaped, and they secrete mucus. Mucus can have enzymes in them, and have all sorts of different things in them. So we find goblet cells lining in the lungs, lining in the nose. So to get all that mucus, we also have mucus in our small intestine, and so we have goblet cells there. Our blood and lymphatic capillaries are all important in removing digestive food. So our lymphatic vessel is attached to the lacteal, which obviously is where our fats go. So we have a large number of mucus secreting goblet cells helping the passage of food. That's really useful. It means that it basically just keeps it fluid and moving. It means nothing's going to get clogged up and stuck and just go really horrible. The tips of the villi are actually constantly damaged by the passage of food. If you think it's almost like as if food's coming past and you just rubbed your hand every time food was coming past, like your skin cells would get damaged a lot. The top of the villi are damaged a lot. So these are easily replaced by cells from the base of the villi. And actually what happens is these cells migrate to the surface and gradually move up the villus until being shed from the tip. And this takes about 5 to 7 days. It's not really how we would normally replace cells. Cells often, like if you cut yourself, for example, the cells replace in that same space through mitosis. And though mitosis will be happening, it happens in a different place and moves upwards because they're going to need replacing constantly. It's sort of a similar way, I guess, to your nails, but we don't shed these, we have to cut them ourselves. But yeah, so it basically moves up from the bottom bed all the way up to the top. So I guess, yeah, so it is sort of a similar way as fingernails and hair. So the difference between a villi and a microvilli. So a single villi within our small intestine, well, that will be made up of intestinal epithelial cells, and then each of these cells will have multiple hair-like projections from the cell with a microvilli. So to describe how the diffusion gradient is maintained across the villus, this is a question that gets asked a lot. So the capillary network is really closely associated with the villi. And so then anything absorbed is quickly transported away to continually provide a low concentration of absorbed substances in that capillary. So that's the idea I was saying, where we'd hit equilibrium too soon and then we wouldn't have any movement. And then to describe the roles of the lymphatic vessels, and that is just to absorb fats from the small intestines. So fats, fatty acids and monoglycerides, they get emulsified, they get turned into micelles, and then fatty acids and monoglycerides leave micelles and enter the epithelial cell via diffusion. The fatty acids link together to form triglycerides, and the fatty globules combine with proteins to form chylomicrons, and this is inside our Golgi apparatus. As you can see, we've got vesicles being formed there. So the chylomicrons are extruded from the epithelial cell and then enter a lacteal, the lymph capillary, and we do this by exocytosis. So we join a vesicle, they are extruded, and then they go into the lacteal here. And the lymph in the lacteal transports chylomicrons away from the intestine. Again, here's another image. So we have fat globules and lipids, and we have bile salts. Those bile salts will help emulsify those fat globules, digested by lipases. So then we should have fatty acids and bile salts. And then we form micelles here with the bile salts and the free fatty acids, so that then it can be taken to the epithelial cells. Fats can be absorbed via diffusion, and bile salts then get distributed and reused. So one, monoglycerides and fatty acids associate with bile salts. Micelles are 5 nanometers, and they are emulsified lipid droplets. Micelles are then broken down when they can come into contact with the epithelial cells. This then releases the monoglycerides and fatty acids, which can then go and diffuse across the membrane. So absorption of fats again. So then inside the epithelial cells, the monoglycerides and fatty acids are transported to the endoplasmic reticulum, and from there into the Golgi apparatus, where they are recombined to form triglycerides. So it's a bit of a strange one. We break them down from triglycerides to then reform them as triglycerides. It's strange, I know. So the triglycerides combine with cholesterol and lipoproteins to form structures called chylomicrons, which are particles adapted for transport of lipids. Basically, to get into the lacteal, the triglycerides would need to pass through the cell. To be able to pass through the cell, they need to pass through the cell as monoglycerides and fatty acids, and then can get reformed in the Golgi as triglycerides. And 5, our chylomicrons move out of epithelial cells by exocytosis, where they then enter the lymphatic capillaries called lacteals, found in the center of each villus. So what causes fat globules to emulsify? Well, like we said, they combine with biosalts to form an emulsification, which then allows our fats to be swollen for digestion by lipases into fatty acids and glycerol. What is the role of Golgi apparatus in lipid digestion, not liquid? Well, Golgi is actually where the monoglycerides and fatty acids then reconstitute into triglycerides. And then finally, what are chylomicrons and what is their role in fat absorption? The chylomicrons are triglycerides that have associated with cholesterol and lipoproteins. These form a structure that enables the triglycerides to leave the cell via exocytosis. An exam question, 3 marks. Explain the advantages of lipid droplet and micelle formation. So, why are droplets important? Droplets are great because they increase our surface area. They increase our surface area for enzyme action, for lipase to attach. Therefore, if we've got an increased surface area, we have faster hydrolysis or digestion, whichever way you want to describe that. Then we go on and say, well, what is the benefit of micelles? Well, micelles then carry the fatty acids and glycerols, fatty acids and monoglycerides, however you want to say it, to and through the membrane into the epithelial cell. So we actually need in those micelles to carry all those parts through. We need all of those parts because obviously they'll then reform into triglycerides inside the cell. You cannot have micelles increase surface area. You can't use the words breakdown. It's a weird one. We don't like the term breakdown because it's very woolly. It's not really succinct and clear in science. We like things like hydrolysis, digestion, not breaking down. Again, the same with things like small enough. We don't like terms like that. We want to be really precise. Again, name structure Q in the diagram above and suggest how it's involved in the absorption of lipids. Okay, so Q, we've got the Golgi apparatus. What does it do? Well, it modifies, it processes triglycerides. What does it do? It then combines our triglycerides with proteins, and then they are packaged for release exocytosis, or you can say they form vesicles. You can't have synthesized triglycerides, and this is quite important because they're not making triglycerides, and it's really fine detail. So, synthesis, mitochondria is a site of ATP synthesis, site of respiration, and ATP synthesized there. We're just reforming and we're processing triglycerides there. So we're getting constituents going to it. If they've already been triglycerides, we've already had to take in triglycerides, then we're forming them. If we're going to synthesize them, it would mean that we wouldn't have to eat them, but obviously we do. So that's why we use the word modifies processes rather than synthesizes triglycerides. And it's quite a small difference, but it's really important so that you can get those marks. And you understand the difference between them. And it's just the idea that to make those triglycerides, we have to already take in those triglycerides. We're just breaking them down. Oh, not breaking them down. We are hydrolyzing them to then reform them. So we're modifying them. And we've got another question here. Explain the importance of structures labeled P. I can't see P. Okay, got microvilli. Why are microvilli important? Well, they increase the surface area for absorption. Name R and S. So obviously S, I'm assuming we're talking about a blood vessel, a capillary, and R will be our intermembranous space. So tissue fluid, interstitial fluid, extracellular fluid, intercellular space. S, lymphatic vessel, vessel. O, lymph, capillary, or lacteal. To be fair, I probably should have read the top question. Oh yes, sorry. So resembles triglycerides going to the organelle. So yes, it would be a lymph capillary. I am going to pause here, and what we will do is we'll finish this off tomorrow. So we'll go pick up from here tomorrow, and then further on tomorrow, what will we do? I'm trying to think now off the top of my head.